welcome back to another microbiology lecture. This one we continue our dive into the structures that we find in prokaryotic cells in greater detail. So again, when I have our foundation prokaryotic cell is very accurate, but when we get into the details, it gets a little bit more interesting. And whereas the first one looked at the plasma membrane differences, let's look at the cell wall. So we'll do the cell wall in this one, and then we'll do one more on external structures and internal structures. So the cell wall, more than just peptidoglycan, and then we're going to emphasize also some of the differences in greater detail between gram-positive and gram-negative cells. So peptidoglycan, as we memorize the different kinds of cell wall materials, kind of our mantra that we learn in intro is that plant cells have cell walls made of cellulose. Again, take a plant biology class and you'll find out that they're more complicated than that, but that is our kind of base layer. We talk about fungal cell walls being made of chitin, and then bacterial cell walls being made of peptidoglycan. So let's dive into that base material peptidoglycan first, and then look at the differences between both gram-negative and gram-positive. So if we look at this diagram, at the bottom you kind of see these repeating strands of glycans, right? So sugars. So we see the alternating NAMs and NAGs. So N-acetylmuramic acid and N-acetylglucosamine are going to be our major kind of strand that we're going to use, that we're going to weave this cell wall with. And again, we want to emphasize the idea here that cell walls are not walls per se. So when I say cell wall, I think chain link fence, and I'm going to kind of use that picture, that image just a little bit later here. Not a stone wall, not a wall in your house. Because when I look at a wall in your house, I can't see through it. I can't toss a glass of water through it. Air doesn't move through it. Where the cell walls that we find in bacteria, plants, and fungi all are much more like that chain link fence. It's a weave of polymers that is basically creating an environment that molecules readily diffuse through. It's mostly water. Anything that's small enough will just go through it like it's not even there. So we want to remember that. And so the main strands that we're seeing here in peptidoglycan are these glycan strands, but the name in fact implies there's a protein component, the peptidoglycan, the peptide. So the cross-linkers to those strands are going to be what we call a tetrapeptide. So again, fortunately, here we have a situation that is somewhat rare in biology, where the names make sense. Peptidoglycan, protein, sugar. Tetrapeptide, 4 peptides. So it's a 4 amino acid structure that's going to link these larger strands together. And that's going to create strength and continuity with kind of how the whole thing holds together by those cross-linkers. Now, this is one place where we'll find D-optical amino acids. I've done a short lecture on that, but just know that it's very unusual to find D-type amino acids in nature, because almost everything else, when we think about kind of typical transcription, translation, enzymes, the proteins that do stuff in your cells, those are all going to be L. Almost all the things that we look at in life-based chemistry are L-optical, and so it's unusual, so it's worth kind of pointing out. Now, when you have a gram-negative cell, you have a cell wall that uses a direct tetrapeptide crosslink between the two glycan strands. When you have a gram-positive cell, there's a special interbridge that we can see here in the middle of this diagram in between the tetrapeptides, right? So that's a little extra kicker in there into the structure when you're gram-positive. So let's definitely now take a look at gram-positive versus gram-negative. So this is now more typical to, again, a review level. We threw a little teichoic acid in there on the gram-positive, but we'll get to that in a second. What you may remember is that when we see a gram-positive bacteria, and I like this picture because it kind of shows you the cell in a cross-section cutaway. As I dig down into that cell, I hit the plasma membrane. But we see that whopping cell wall, like boom! There's a mass of peptidoglycan, and then there's the plasma membrane. But remember, that's a weave, right? That basically is permeable to most things, and that it's really the plasma membrane that's dictating the chemistry of the cell, that's maintaining the integrity of the life chemistry, the gradients that are important. Now we contrast the gram-positive to the gram-negative, and now if again moving from the inside out, we hear plasma membrane, we hit a peptidoglycan cell wall. It's not nearly as thick, and then we hit another membrane. And so this does create a space that we're going to talk about in a second called the periplasm, which is usable by the cell as a compartment, right? So already we're starting to kind of break down that idea. It's like, oh, prokaryotic cells only have one space, but if you're gram-negative, you kind of have another space to play with, the periplasm. But let's dive into the gram-positive strains first and look at that honking thick peptidoglycan cell wall. All right, so here's another visualization of that. And so as many as 30 glycan layers are there, which is significant, right? So that's going to build up some thickness. Now, what I think is interesting when we start to dive into more detail is the fact that teichoic acids are woven in between these strands. And this class of chemicals is extensive. It's up to 60% of the cell wall mass. So these teichoic acids are attached to the membrane and to the cell wall material itself, to the peptidoglycan. So we talk about lipoteichoic acids, and that totally makes great sense because now all of a sudden you have a covalent bond, a molecule that's anchoring the cell wall to the plasma membrane, right? Now some gram-positive bacteria will even have a tiny space in there, a periplasm per se, as it's named, but it's not nearly as extensive as we'll see in the gram-negative bacteria. And it's almost non-existent in many, so it's kind of sometimes there, sometimes not. We'll also see teichoic acids attached to the cell wall, and those will extend above and beyond the peptidoglycan layers. So these are going to be important, as you'll see in the next couple slides, for the interactions between the cell and its environment. So the chemistry of those teichoic acids, as well as the peptidoglycan, are going to be what are touching the environment, and that always is going to play a role. So what I like to think of this analogously is a chain link fence with vines growing in it. Right? So you can see kind of pictures here on the left, like there's clearly chain link fence, and you can see these vines starting to grow and they're interweaving between those spaces. But the chain link fence is kind of the main structure that's supporting everything. And then on the right, you can see how that can like really blossom out, right? So again, the chain link fence, your peptidoglycan layer is your kind of major support network, but I can expand the stuff that's kind of interwoven it quite extensively. So this is kind of the picture I see in my head when I think about those teichoic acids. Now, when you get into more detail, and you can look up reviews on this stuff, here's one I pulled from a review from 2008. Great picture, kind of looking at the importance of these teichoic acids in interactions with structures that the bacteria will touch, right? So physical chemical surface properties are going to affect how bacteria stick to stuff. So specifically when we think about medicine, we think about bacterial growth in catheters. How do phage viruses interact with bacteria? They're going to be interacting with those teichoic acids. Host recognition, again, with our immune system. But even beyond that, then we're going to think about how those teichoic acids are influencing the prokaryotic cell of which they are on. So they can also act as a reservoir for cations. So it can be storage of, we see magnesium here and hydrogen ions, and that then becomes physiologically significant, right? So it's not a true compartment, but it's almost is in the sense that I have such a negative charge that I can store a bunch of positive charge and then draw from that as needed. So there's more going on there. Now, so when we kind of view this from a different point of view, again, I like this picture because it's just really clean. From the side, you can see the cell wall and the gram-positive. And here on the right, you can see that periplasm, that gap between the plasma membrane and the start of the cell wall in now what's a gram-negative bacteria. So when we look at that gram-negative bacteria now in more detail, we want to focus on that outer membrane, right? So we have a thin cell wall in gram-negative bacteria, and they're devoid of those teichoic acids. So those are exclusive to the gram-positive bacteria. Now, the outer membrane is bound then to that cell wall by lipoproteins. So again, lipoprotein being a general name for a protein that has covalently bound lipids to it, and that's going to act as an anchor for a protein into a lipid bilayer. In this case, what's really significant about most gram-negative cells is the fact that that outer membrane has two different leaflets. And the outer leaflet is very significantly known as a lipopolysaccharide, or LPS. Why do we care about that so much? Because it's going to have a real big impact on our immune system, which we'll talk about in more detail later in the quarter. But for now, it is an external membrane, right? It's an effective barrier, but it also then has to have some controlled permeability. And we see a major class of proteins in that outer membrane called porins. So it's called a porin because it's a pore-like integral membrane protein. And so those can be specific or more general, like any other channel protein. And then there's the periplasm. So the periplasm then is that space, which again, thinking of the cell wall as kind of this chain link fence kind of thing. It's not a barrier for all these molecules that are diffusing. We have a periplasm then that is a functional compartment, and we'll find various enzymes in that periplasmic space. It can be significant, up to 40% of the cell volume, which is no joke. That's real. That's real. Now, the lipopolysaccharides are interesting because they're a MAMP, aka PAMP. And like I said, we'll get into this more when we get into interactions between microbes and their potential hosts, i.e. us. So it's an innate immune response, a trigger. So a microbe-associated molecular pattern. So this is something that our body doesn't have to learn to recognize as bad. If you were to inject this into any naive immune system, it would immediately say, this is bad, and respond aggressively. And that makes sense from an evolutionary point of view. So it's a kind of pathogen-associated molecular pattern. Plants do this as well as animals. So the idea of MAMPs and PAMPs are really very interesting if you get into the details, because we are being assaulted all times by other kinds of life, as is every other kind of life, right? So it would make sense that things that can be potentially very harmful, that you would have a very, very significant trigger for that. So it's considered an endotoxin because if it gets inside you, like into your bloodstream, if it's a sufficient amount, like literally your immune system will kill you because you'll go into a shock response, because it's like, this is bad. There are bad bacteria here. So the lipopolysaccharides are made of two parts. You have the lipid A protein, which is the part that's like really dangerous to your immune system. And then you have this inner core, outer core O antigen repeat. And so this is where the variability is going to be found from species to species, right? Okay. So we bring this up, and it's important to think about the cell walls and specifically peptidoglycan because it's something that we do not have, right? And is essential for the bacteria. Again, think about the osmotic problem. When you have organisms living in hypotonic solutions, which most solutions that bacteria live in, think about ponds, liquids in general, water is going to want to move into the cell. And if water moves into a cell that doesn't have structural integrity, the cell will ultimately burst. That's why it's so important that we have isotonic solutions in our bodies, because our cells don't have cell walls. So that cell wall is integral. If you have a faulty cell wall in a bacterium, ultimately it's going to burst and die. And so if we can attack the integrity of that cell wall, as many organisms out there do with their antibiotics, you can kill bacteria. And that's what we've learned. So and we'll talk more about antibiotics later in the quarter as well, but it's a great target. So the illins, penicillin, our first antibiotic, prevents the cross-linking of the glycan chains. It's an enzyme inhibitor. So again, when I'm constructing the cell wall, I have enzymes out there that are doing the job of cross-linking those glycan chains. And if they don't get to do their job, then the cell wall doesn't have integrity and it falls apart. So the whole family of illins, and we'll talk about this more later, some of them are very effective, and they tend to be more effective against gram-positive bacteria, but some are designed to cross the outer membrane, and so they can be effective against gram-negative bacteria as well. The other thing we want to think about is a natural enzyme that we all produce in our tears and many bodily fluids. It's lysozyme. So the lysozyme is an enzyme that breaks the glycan chains between alternating subunits. So again, what's the strategy here? If I can break that cell wall, the cell loses integrity, becomes weak, and it will burst in a hypotonic environment. So super cool, gram positive, gram negative, definitely something to be able to talk about. Let's talk about archaea as we finish things up. Archaeal cell walls are going to be all over the board. So varied, right? So when I say archaeal cell walls, like, ooh, varied, but not peptidoglycan. So some have a pseudopeptidoglycan, and I could probably do a whole lecture on archaeal cell wall structure. Great reviews out there. Start here as I did with the Wikipedia page. But you'll often see what's called an S layer associated with archaeal cell walls. And so I like this quote because it like really resonated with me from this article. A two-dimensional macromolecular crystalline structure formed by sometimes glycoprotein subunits that cover the surface of many species of bacteria and almost all archaea. Right, so you can start to see here gram positive, gram negative bacteria. There's that familiar structure. You can see plasma membrane, cell wall, and then here we represent that S layer as these kind of yellow proteins kind of covering the surface. So again, a lot of this is about exposure to words, and when these come up in the future, you'll be like, oh, I've heard of that. I can learn more about it. So when you think about archaeal cell walls, like be like, yes, they're different, no peptidoglycan, variable, S layer. There you go. Finally, we're going to end up with some bacteria with no cell walls, right? So obviously they're going to have other strategies to control osmolarity and deal with the osmotic problem. But mycoplasma is like the big thing. So this is what we're going to worry about. One, because a Mycoplasma pneumoniae can cause walking pneumonia. And if you don't have a cell wall, all of a sudden all your antibiotics that are working at attacking the cell wall aren't going to work on mycoplasma, right? So their membranes are going to be modified, increased sterile concentration to improve that membrane strength. But again, I'm more concerned about this because if I get an infection, I have a whole class of antibiotics that aren't going to work. So there's exceptions to everything. Mycoplasma is also one of the big things to worry about if you're doing tissue culture with mammalian tissues, because if you get an infection, if you get a contamination with mycoplasma, it's really hard to get rid of. Again, because it tends to be resistant to most of the common antibiotics that we use as selection factors. So with that, then we go back to our summary slide. And now we can say, okay, look, we've done a quick summary of the cytoplasmic membrane, and now cell walls, gram positive, gram negative, talked about archaea a little bit. So I'll go on to our final lecture on the details of the prokaryotic cells and look at external structures and internal structures, just so we've had that exposure.